Welcome to our third video on NIH grants and NIH style grants. And in this video we'll be focusing on the significance and innovation review criteria. This is the third of a four-part video sequence on writing a research plan, and again we use the current 12-page NIH format. Here we're going to focus on the significance and innovation sections using the grant-winning examples. So in this video and other videos in this sequence, we analyze two winning grant applications available from the NIAID website. These are the Parrish and Ratner applications. You'll find the link to the website here. You can also find PDFs of these research plans at the CLIMB website. Note, to take full advantage of this and other videos in this sequence, you should have read the research plan of both applications before you view this video. We're dealing with a lot of important technical information, and it's important to have a basic understanding of what these applications are trying to accomplish. Now, let's match the NIH review significance criteria to Parrish's discussions of significance. Recall in our previous video how we outlined what those were. Does the research address an important problem? And Parrish is very specific about the importance of the problem, understanding fundamental aspects of virus infection processes. Critical barrier. Again, explicit addressing that. The critical barrier to developing effective antiviral therapy and vaccination success. Our lack of knowledge is an impediment to that. And we have an improvement in knowledge. We'll understand the fundamental viral mechanisms and clarifying the textbook knowledge of virus structures and function. So we're ticking off those criteria here in the significance section. Now, specific text from Parrish aligns to these important problem criteria. The adeno-associated viruses are parvoviruses that are not associated with disease but are being developed as human gene therapy vectors. And the same issues of receptor and antibody recognition are important for vector optimization. You can see the importance of that problem, how this study will help us in this area. Another important problem that the study addresses, variant viruses with extended host ranges can cause new outbreaks or epidemics of infectious disease. Hey, nothing more important than that. The viruses that we are examining include the comparison of such a system where one variant arose as a pandemic pathogen in a new host through the acquisition of mutations in the capsid protein, that component that they're studying, that altered its structure to change host-specific receptor binding and also to change its antigenic structure. And now specific text from Parrish aligns to the critical barrier criteria. There it is. A better understanding of the details of general processes involved will likely allow the development of more effective and broadly acting antiviral drugs. Although antibodies are critical components of immune responses of all vertebrates, in many cases they are poorly effective so that viruses maintain persistent infections or vaccines do not work well. Understanding the underlying rules that determine how antibodies bind to viruses and block the processes of cell infection will reveal how effective antibody responses might be elicited against different viruses. And then finally, specific text aligns to improvement in knowledge. Here we'll investigate the roles of flexible and variable structures in the parvovirus capsid and show how receptor and antibody binding control cell infection. We're advancing the knowledge in the field. Again, the significance section explicitly connects up to the important criteria. Now let's match the NIH review significance to criteria to Ratner's discussion of significance. Recall our significance section with its 8 paragraphs and its logical flow. We have an important problem. The size and cost of the problem is significant. We have an improvement in knowledge. The pathogenesis of this particular bacteria is not well understood, and we have a novel approach to understanding it. And then we have a critical barrier. The critical barrier is, up to this point, we haven't had an animal model that we can actually use to explain the pathogenesis. And specific text from Ratner aligns to the important problem and knowledge criteria. Here's our important problem. Bacterial vaginosis among reproductive aged women are roughly 30% of overall, corresponding to 21 million women with this affliction. And rates are higher in pregnant women and in African American populations. It's estimated there are 90,000 excess preterm births per year. And the cost of this, over $1 billion. Plus, women with bacterial vaginosis are at increased risk of acquiring sexually transmitted diseases. The importance is clear. There's another importance too. The particular toxin that they are investigating, VLY, it provides an opportunity to expand knowledge about that whole group of toxins, not just this specific one that they are looking at. Note too that it increases our knowledge. It's an important problem, but knowledge is also related to that. We're going to know more about this specific set of toxins when we look at this specific toxin. Now, specific text from Parrish also aligns to the critical barrier criteria. And there it is. By altering both bacteria and host, we will produce murine models that can be colonized by a variety of G. vaginalis isolates, and will construct G. vaginalis strains that can colonize a wide range of mouse strains. These two strategies will address the major barrier to understanding the pathogenesis of bacterial vaginosis, the absence of an animal model. Explicit connections to the notion of a critical barrier. Now let's match the NIH Review innovation criteria to Parrish's discussion of innovation. Here, canine and feline viruses will allow for studying complete virus infection model. Now the methods proposed are being used in viral studies. In viral studies, proposed studies include combinations of methods that are being used in novel ways. So the methods themselves aren't necessarily novel, but the combinations are. And we're focused on methodologies. We use a variety of advanced methods and structural biophysical, biochemical, and functional assays in these studies. While many of these methods we propose are being used in viral studies, the proposed studies include combinations of those methods that are being used in novel ways to gain a complete understanding of the mechanisms involved. So let's match the NIH Review innovation criteria to Ratner's discussion of innovation. In that case, the innovation focused on genetically manipulating the microbe and the host to create the new animal models, and their innovative methods to do so. And again, it's a focus on methodologies. Our characterization of a human-specific toxin from G. vaginalis and its receptor on the surface of human cells represents an opportunity to overcome the narrow host range of this bacterial species. By genetically manipulating both microbe and host, we will create new animal models for BV and understand the specific role of G. vaginalis in this pathogenesis. We have used innovative methods to generate the first descriptors of genetic manipulation of G. vaginalis, chimeric toxins with altered species specificities, and the first successful model of murine colonization. So you can see how this discussion of innovation, very brief, hits on some key points related to innovations and methodologies. As you write your proposals, make sure you hit the key touchstones in the review criteria. With significance, The most important thing is, does your research address an important problem or critical barrier? And connect up those concepts explicitly in your discussion. Will you get improvement in knowledge or technical capability or clinical practice? Again, connect your discussion explicitly to these criteria. And for innovation, again, look at the review criteria and make sure that you connect up your discussion with the criteria that apply in terms of innovation for your proposal. If you make those connections as direct, as clear, and as explicit as possible, your proposal is likely to be much more successful.